Great. I think the webinar is filling up. Yeah. So welcome everyone. And yeah, welcome to the first acib webinar on modeling and simulation. Yeah, the reason we have this webinar is, I mean, historically, acib has been very strong in using, has a strong history in using modeling and simulation as a tool to optimize or automate processes or to answer scientific questions. And we figured that we want to use this webinar to showcase our capabilities at acib and also at partnering universities and companies and really, yeah, give everyone an idea of what expertise we have. And yeah, it's my pleasure today to have speakers from Novasign, a company that has had a long history with acib, and also BOKU University, one of the partnering universities of acib. And today we will talk about mechanistic and hybrid modeling in downstream processing, and some organizational information about the webinar and how we will structure it. So first, Dr. Krippl will give a presentation of 20 minutes, then we have 10 minutes for questions and answers, yeah. And then Dr. Beck will present for 20 minutes and then 10 minutes of Q&A. As you see, we will record the meeting, yeah, but the Q&A, you can ask questions also anonymously if you don't want to be identified. And we will stop the recording after 1 hour, and then we can continue the discussion if there's interest. Yeah? Okay, so without further ado, yeah, it's my pleasure to introduce Dr. Maximilian Krippl of the company Novasign. And yeah, please, you have the floor.
Thanks, Nico. I think you can all see my screen. So,
looking great.
Perfect. Well, thanks for the kind intro. My name is Maximilian Krippl. I work for the company Novasign. Started my career at BOKU University. A big part of what I'm showing today was actually done in collaboration. And yeah, very excited to kick off this webinar series. So I'm working for Novasign, and Novasign is a modeling company in the field of bioprocessing. And I just want to start out with a small vision with a slide of the vision of what we're actually doing and what we want to achieve. So everything we're doing is focused on generating useful models for partners and for customers. And they normally have 3 pillars. So one thing is we need experiment. We all know that we need experiment in order to make useful useful conclusions, and that's one side, to really generate smart experiments. So a small number of experiments, but with a good predictive power in order to be able to learn from them. So that's one big part, the smart experiments. Then we need a way to aggregate all of this knowledge, data, into a single platform. That's why we developed our own software solution in order to bring all of this information together. And the third pillar is actually the application. So we want to use, based on the smart experiments and process understanding, we want to use these models to make meaningful predictions. So where is the optimum of my process? How does my process look when I scale it up? How can I apply this in upstream and downstream? A small history. Novasign was founded in 2019, a spin-off of the BOKU University. The idea was actually born already earlier, so in 2017. At that point, modeling generally was, let's say, more an academic exercise. There was not many applications in industry, and we thought with what we managed so far, or with what in 2007 we done already, actually we could start a company with that and sell these solutions to industry and then help industry to advance in this field. And over the years, we started actually with upstream, with microbial upstream process. We generated more and more use cases. So first of all, we went into downstream. We saw that downstream was an untapped territory, especially filtration. This is what I'm going to talk about today. But later on also we tapped into ATMP, biopharma, industrial biotech, also cultured food. And over the years, we generated, with more and more projects, we generated more and more knowledge that we brought together just last year. We finalized our what we call process engine. So all the knowledge that we have from our processes that we modeled so far is in there so that we can quickly build new models for new processes. In addition, we also have an assistant that helps to basically set up this process so that it no longer remains an expert tool, but so that more and more people that actually work in the lab can use it and can generate useful insights. A couple of words to this. So the objective here is really to create a full process chain of my lab that represents my lab process, like my fermenter, my filtration, my chromatography device, and build models with pre-configured block. And one of this pre-configured block, or the journey to that pre-configured block, so the filtration, this is what I'm going to show today. It is accompanied, we want to achieve that with not more experiment, but ideally less experiment. So we worked hard on setting up smart experimental planning to reduce the experimental effort. One big aspect is scaling up and transfer of processes. So how does my process behave when I go from the lab scale to the pilot scale? And here we saw that our original models we started with in 2017 are not fit. Therefore, we let's say, accompanied them with CFD models in order to allow for better predictions. If you have a good process model that predicts the performance, you can quite easily adapt economic factors to it. So you can do a cost of good analysis on these processes as well. And something that we want to do, but that is not really there yet, but what our models are already capable of, is to have a self-controlling process. So if you know what's going on in your process and your model can quantify that, we can also find automatically strategies in order to mitigate potential risk or batch failures. And the idea of this platform is that it is a fully transparent platform that people can use or plug in and plug out whenever they want. If they say if you don't have a process model, you can try the solution here and then create your own process model. If you have one, for example, you can load it in here, for example, Python models, and then you can make use of the simulation environment in order to optimize the process, for example. At the heart of all of this, and this already comes closer to what we're talking about today, is our hybrid models. So hybrid models, simply said, are a combination of machine learning and mechanistic models. And for us, the big advantage of those models is that they're very flexible. So the inputs and outputs of the models can be adapted to use cases, which is a limitation of some mechanistic models. But the advantage is that you need less data than if you would use a pure machine learning model, like a regression model, for example. And that makes it actually perfect, or a good sweet spot, especially in the field of biotech, where we have data, but not tons of data, like, for example, in social media, where we have tons of data. So here we really have every measurement we do is very valuable. And the advantage, since you can change the inputs and outputs, you can put them together and describe multiple steps of one process. So for example, you can then concatenate a reactor with a filtration and see what effects do changes in temperature, for example, during the cultivation have on my filtration. Okay, let's delve into the showcase of today. So ultra-dia and SPIT, ultrafiltration, diafiltration, and single pass tangential flow filtration. So this is a step that is commonly used in probably all processes when the goal is to concentrate and to buffer exchange the product. And what we saw from the very beginning is that ultrafiltration generally was not really aim of any modeling exercise. So we saw a lot of experiments go into the optimization of the upstream process, into chromatography, and filtration was normally always, yeah, we have a similar product, we take the same conditions, and it will work out. But actually, we found a way that with few experiments, you can make quite some significant adjustments and optimization to that process. And the questions we asked ourselves when we started this a couple of years back, and that we also then were reinforced by the questions of our customers were actually the same three, more or less. So you had what kind of membrane should I use for this specific task? Then how should I operate it once I have my membrane? What pressures, what flows? How should I set up my ultra-dry filtration? And last but not least, if I have this process now set up in batch, how should I do it what should I change in order to accommodate a single pass or the continuous version of it? And of course, how can I do this without needing a lot of experiments? And this is what I want to start with, this requiring few experiments, because here we have a very good way of actually testing different combinations. So something that was not developed by us, that existed before, but which we say used to or pushed to the limit and then used to a big extent, were parameter scoutings. So in filtration, the advantage is that you can, by recirculating your solution, you can test with a small amount of product or intermediate, you can test your system. So in this case, we tested here different transmembrane pressures that we applied to the system, different flow rates, and different concentrations. And we saw what effect this has on the permeate flux, shown here on the y-axis. Let me just take the pointer here. With this, we are able to cover actually the entire operational space that a device in lab or later on also pilot and manufacturing scale is capable of covering. And then, this is then part of the modeling. How can we apply this now to different scenarios? The easiest way, a batch TFF, which is the simple TFF you might know from the lab. You concentrate your solution and basically take it out and bring it to the next processing step. A fed batch filtration, so where you have larger volumes. A diafiltration, where you actually do buffer exchange. So you replace the existing buffer with a new buffer that is better suited for formulation. And then also the single pass TFF, so the continuous version of it. And actually, this here is the most technical slide, but I just want to give you a flavor what it means or how in a hybrid model can look from the inside. Yeah, this is a very simple or the simplest example that we came up at the very beginning. So this is the batch ultrafiltration model, and here we try to focus on the parameters that we can control at any time and that we can measure quite easily. So what we saw mostly, what affects our filtration is the transmembrane pressure, the feed flow, and the concentration of our product or products. And they influence our permeate flux, so how much volume I get out of my system. And all of the complexity, so basically things like buildup of concentrations or potential fouling or potential clogging of membranes, this is then covered here in our neural network, in our ANN, which is our regression part of the hybrid model. So this takes care of the complexity that also changes if I take another product or another membrane that I can a priori not really predict. And then I just feed it into this quite simple mass balance, which tells me how do I, what under the current circumstances, what does it mean for my process if I now predict with this flux and the membrane area I have, this A here, and my current concentration, what does it mean for the concentration buildup? The goal of ultrafiltration is to increase concentration and to reach a certain level of concentration. And now I can actually do this in a loop. So I take this concentration here on the left, on the right, feed it back into the left, and then I perform this until I reach my final goal, for example, a final concentration or a final volume that I want to achieve. And what the model gives me then is how long the process takes and additional performance parameters. So I want to show you a showcase here. We had an ultra-dia ultrafiltration that we trained our model on, and the scenarios we wanted to achieve 150 grams per liter. This is, for example, for a monoclonal antibody, a good example. Now, when we developed this a couple of years back, 150 grams per liter were okay. Now, when you look at the publications, you could argue it's actually a bit low. You want to go even higher, like 200 or even more. But the question is, when should I actually stop? my ultrafiltration. And here we did some runs where we say, okay, what happens if I just go until 50 grams per liter? So I concentrate up to 50. I think we started with 20 grams per liter. We go up to 50, then do always the same 10-fold buffer exchange, and then increase finally to 150 grams per liter. And we see this process takes roughly 160 minutes. We repeated that. We just changed the set point here. We now say, okay, go until 150 and then do the rest of the diafiltration. Then we don't need the second ultrafiltration. And that's already much better. Okay, I could say clear, that makes sense. You exchange less buffer. You obviously need less time. But interestingly, when you simulated 110, you'll see that you're actually even faster. So meaning there is some kind of sweet spot in there between buffer volume and membrane or concentration buildup on the membrane that increases the resistance. And then we can use the same model just in a different setting and just simulate for different concentrations of ultrafiltration. After ultrafiltration, how long does my process take? And actually, you see here, we're roughly at 120 grams per liter for this specific membrane that we used, and this specific product seems to yield the optimal results. Below, we are exchanging too much volume, and above, we are simply the membrane, you can say, is clogged or starting to clog. And since this is very different for every membrane protein or product combination, we really focused on reducing the number of experiments here, because you cannot really, or we didn't find a way to really transfer this learning from one membrane to the other. And with this approach also, I think it's not necessary. And one thing we really focused on in the last years is to bring this into a solution that is really where you have everything in one place. So every graph that you just saw, we can generate within the same, so that's the same process or the same software in the end. We have here the ultrafiltration, and we see that if we just, here this is the view that I just showed you. So the duration of the process over the concentration of the diafiltration. We see that by changing our feed flow and transmembrane pressure, the optimum changes actually quite a lot. Here we have our ultrafiltration, and here we have our recovery model, because this was actually something that we found out in a discussion with the customer, saying, okay, it's nice that we can actually model the highest concentration, but how do we get it out of there? It doesn't help us anything if it's just in the device and everything is stuck to the membrane. We need a recovery strategy. And here I want to mention some highlights why hybrid modeling is so, let's say, adequate for this approach, is because you can extend it. So, for example, here in the middle, we have for the diafiltrations that we did an pH model. So everything I talked about until now was mainly performance. So how do I get it faster and reduce, let's say, time of or utilization time, but actually quality-wise, this does not really matter. You can run, you can also need more time. It's just you need to achieve certain criteria, like for example the pH at the end or the excipient concentration at the end. This you have to reach, and we can extend the model with a pH model, for example. The one I'm showing here is again a machine learning based pH model, but it can, for example, also be a mechanistic one. Based on the Donan effect, for example. And here it really depends, and as we found out, it really depends on the collaboration partner and the customer which approach to take, because some of them have preferences for one and others have preferences for the other. Sometimes speed is more important than knowledge, and sometimes knowledge is more important than speed. And here on the right, you can see the same thing for the SPTFF. So we have here the scenario with 1, 2, or 3 membranes in parallel, and we wanted to see if the model can actually capture the trend. We can actually see quite well what feed flow, for example, means for different conversion rates. So higher conversion rates means higher concentration. And we validated this model also with the dots you can see here. Overall, for the process we simulated here, we saved 2 weeks of lab experiments, so really meaning experiments where you would test high pressures, low pressures, high flows, low flows, and so on. And here we can cover everything within one run. And the final run was 18% faster than what we started with. Here just a simulate, or basically I want to show you how much experiments we actually need in the beginning, how many we need. In the beginning, we started with 390 observations. You have to think through, like we always set the parameters, the pressure, the concentration, and the flow for like 3-4 minutes, keep it constant. So 390 actually takes quite a lot of time. This took 3-4 days in order to capture all of this. And we got an error of 1.5 approximately. But we saw if we reduce first by 80% these points and later by 90% these points, we actually get the same result. So we were way overshooting the number of experiments that we needed. And we actually saw with just around 40 observations, this can be done within a day. So this is an experiment that takes 5-6 hours. We can really cover everything. Okay, this is everything I wanted to say about filtration. I just want to mention, since this is an acib webinar, I wanted to mention some collaborations where we already applied our technology or are applying our technology successfully. So one of these projects is the CyanoCare project. This is done in collaboration with acib and BOKU. So the project lead here is Rita Mota, working together with her PhD student, Daniela Vucic. And we work here with our partner Simvantage, especially with Lucas, who's an expert in CFD modeling. And these are now upstream projects, so the scope is different, but the modeling approach is actually very similar. And here the objective was to optimize the cultivation condition for cyanoflan. Cyanoflan, which is a polysaccharide, which has skin protecting attributes, and to be able to produce this cost efficiently in industrial scale for the potential use in skincare products. And here we started what the pressure and the concentration were before. This is here now light intensity, because this is a light sensitive organism, temperature and the steering of our of the reactor. And we generated experiments here. And based on this experiment, we saw what effects do this parameter have on our biomass, so the OD in this case, and also our cyanoflan, so the released polysaccharide here shown in RPS. So this is a 2-year project. It's ongoing. We're currently in the stage of finalizing the optimization and really going towards the scale up of this process, which is very unique and challenging and here we're working with Lucas a lot in order to really capture what does light intensities from different areas, how do they actually affect my cells if I am in my small scale, which is a regular reactor, and then in the large scale, which is a photoreactor, and potentially even in manufacturing scale where it would happen outdoors. And another project is the eConti project. This is an FFG-funded project where the goal is to fully continuously produce a protein A. So this is a step actually of multiple processes. We have 2 reactors where we build up biomass of our microbial cells, of our E. coli, produce our protein A, and then continuously bring this to a microfiltration where we filter out the cell debris and really just keep the protein A and then put it on the MCC. And this is a project where we're working quite a lot of partners. So on the modeling side, it's again Simvantage and us. On the data capture side and integration side, it's Cubicon. And on the experimental side, it's the companies Engine and the BOKU University together with the company Toso Bioscience. And here I want to show you what these models actually in the right context can actually show you. So on the left, we have, for example, our temperature and pH of our process. And on the right, we have the simulation, the real-time simulation of our biomass and our product. And we can see how in this environment from Cubicon here, we can actually, by changing, you will see now a change on the left side in the temperature and the pH, how this instantly changes our response. Something you would only be able to measure probably a couple of hours or day later. Here you can really have it in real time and decide what experiments to perform in order to improve the product, in this case, the product concentration here shown in yellow. You can see that the combination we have here, we actually increased our conditions or our product concentration quite a lot. Okay, I hope I was able to give you a quick intro on the advantages of hybrid model and generally, especially on downstream processes today. is focusing on TFF. So I showed you that smart experimental scouting is a fast way to cover the experimental space, especially focusing on the optimal conditions in the development, but then also going towards larger scales. These experiments that you did can be extended, for example, to diafiltration and single pass tangential flow filtration. So the knowledge you generated with one experiment is not lost. You can simply extend it with another experiment. Roughly 40 points. So one experiment is enough to really get a good understanding of how the TFF works and where the optimal conditions for the same scale are. And we can extend this. These are some projects we're currently working on in order to scale up this process. So what parameters are important to also mimic the conditions while scale up, to model also other process performance related outputs like productivity or recovery concentration, and also in combination with other projects. So especially when you think about intensification, you are having 2 or 3 different process units, and you have a novel way to integrate them into one to increase yield, to reduce the losses. Then filtration is often one part of it, but it needs to be extended quite drastically. And last but not least, we really, in order to stop it just being an academic exercise, we really bring it to industry, we need to integrate this into a useful software. And our Novasign Studio is basically a way to foster collaborative and modular and extensible work in this field. So with this, I'm at the end. Thank you very much for your attention, and I'm happy to take questions.
Great. Thank you, Max, for this really interesting talk. Are there any questions from the participants? Post them in the chat, or I think you can also raise your hand and then we can talk. But yeah, it's easier to do. Oh yeah, we have one raised hand. Yeah, let me see. You are allowed to talk now, Thiago. You can ask the question. We cannot hear you yet. Unfortunately, yeah, I cannot hear anything. Maybe it's easier if you type in the chat. Okay, in the meantime, we have one question from Beck. How do you manage the scale-up geometry changes? For example, the number of pumps of for TFF between lab and pilot.
Yes, it's a good question. And here, originally we started out, we said we want to know the geometry of the system and mimic the flow behavior. But we saw that this, even if you understand it very well, there is some differences. And actually, when you go to industry and pitch this idea, it's really hard to sell it. So what we went for is to take the membranes and the system you have, and first of all do a calibration run. So then you know how does your flux and your under certain pressures and flows does your membrane behave. And if you do the same standardized experiments on the larger scale, then you already have a good idea on the offsets, or at least on one offset. And this is already a really good starting point where we saw that you can compensate quite a big difference in the performance by simply doing this, by simply having this approach. This can be extended later on, of course.
Okay, so Thiago, let's try again, maybe. So
you hear me now?
Yes, great.
It was a mic problem. Thank you, Maximilian, for the presentation. I really enjoyed because I'm also trying to integrate the hybrid modeling into my mechanistic knowledge. My question was more of a curiosity. When you use neural networks to establish the states, like the constants and the parameters, what is usually your bottleneck? Is it the neural network or the actual models or the mechanistic?
It's a good point. It's I would say the more you go downstream, the more it becomes the mechanistic. In our case, it's more the neural network, so the model identification. There, it can sometimes in the beginning take quite some time in order to find the coefficients or the right ANN settings in order to accommodate the complexity. So to make it as complex as needed, but not too complex so that the model doesn't do weird things at the boundaries. So this is definitely the biggest or a big bottleneck, and we're currently working on this by basically redoing our model identification. Currently, it's really you have to set many things manually, but with now frameworks out there like PyTorch and MLflow, so we work a lot on Python. We're trying to find a way to really preset some conditions and then let, based on the performance metrics, do a big chunk of the work by the software, and then later on just do the fine tuning.
I see. And do you have any sort of rule of thumb when you choose the amount of parameters? Because I assume the more the parameters, the more the computation part.
Yes, so this takes definitely longer. I mean, there isn't many rules of thumb, unfortunately. Otherwise, we would probably also be a lot faster. But I'd say, I mean, the number of inputs that you have and outputs should correlate with somehow the nodes you have. So, for example, for the exercise I showed you now, it doesn't really make sense to include multiple large layers of neurons in a neural network. That doesn't really help. It's more we normally stay with one, maximum two, and yeah, I mean the activation functions and so on. You can play around with that, but that's rather standardized. It's more really the how many nodes you have and how long you train, so how you configure your loss function.
Okay.
Thank you so much.
In the interest of time, I think we should go ahead. So you feel free to still ask questions into the Q&A. Max can answer it here, or Max will also be available afterwards for more questions. So since you said, yeah, the mechanistic portion increases if we go further downstream. Yeah, so let's go to chromatography modeling, where mechanistic models are quite strong. Yeah, so thank you again, Max. And yeah, our next speaker is Dr. Jürgen Beck from BOKU University, and I will hand over right away.
Okay. Hello. Yeah, as Nico said, I'm basically only working with mechanistic modeling in chromatography. I started here. I'm basically a BOKU product. I started as the chromatography modeling at BOKU during my master thesis already. And since we had already a talk from industry, I will just briefly say what's the use of chromatography modeling. In industry, it overlaps pretty much with what Max already said. It's for process control. You have to reduce the experimental effort. Imagine you can calibrate a chromatography model already at the high throughput state as people are doing right now with robocolumns and, for example, hydrophobic interaction isotherms. If you can calibrate this and run a simulation, you can drastically reduce your experimental effort if you can already in silico get an optimal range of your process. Or you can, again, like mentioned before, you can have a digital twin and recreate complete unit operations in silico, just also for process control. Imagine your feed is different a little bit, the concentration is higher. You instantly know, okay, maybe my sample pooling should be different right now after the process. So all of that aside, I am more on the fundamental research side at BOKU. And here I have mostly used chromatography modeling to investigate actual transport phenomena. So interesting things, chromatographic curiosities that we observed in experiments, or compared the idiosyncrasies of materials like membranes or resins for certain materials, and investigated complex binding processes. So we're talking about mechanistics, and this always means not a nice ANN box, but rather more an equation description of the process. So I'll briefly introduce the general rate model of chromatography. So here you see a step input to chromatography column, and then the first important transport is outside of the beads, where there's convection and dispersion. And then you have to pass the barrier into the chromatography particles, and there we're going over the film by diffusion, and inside of the pore there's again diffusion, and only here are we in the binding portion where competitive adsorption takes place. This is where our binding models live. Hopefully we'll get an appropriate one. In this case, this would be an ion exchange reaction. Here you see the yellow protein is, let me just get the laser pointer. Yellow protein is obviously positively charged, so it can bind to the resin, and the other protein is not. So all of these transport processes from the interstitial space onto the pore diffusion level are governed by these equations. So this looks quite scary to some people who never have worked with modeling before, but it's actually good because most of these things you can do either set, like here, this is the velocity. You can actually just input that, and all of the other parameters are either material constants, like the radius of your pores, or they can be experimentally determined, like the void fraction of your column, so the part that is not your stationary phase. Then we have the diffusion coefficients and the film mass transfer coefficients, interparticle porosity. These are all parameters that can be measured. So theoretically, you could arrive at a nice description of your column, which is true for most transport, but the devil is in the details as always. So transport is also not so straightforward, as we will see later. Then once you come to the ligands, there are numerous binding models available and have been published. I don't know how many there are, in excess of 20 for sure. But here we see an SMA description for the ion exchange, where we have the ionic capacity, the charge of the protein, the shielding coefficient, and the adsorption desorption rates. So this should theoretically cover everything you would need for ion exchange. Yeah. We'll see later how that sometimes is not the case, because the mechanistic description, yes, you can describe all the processes going on, but nature can throw a wrench in the in things quite easily. And if you've never done anything with chromatography modeling, just be aware that this is like a very detailed description of the transport processes. But here we have a simple case, because I can always simplify it down by excluding minor contributions like axial diffusion, and you can go to a very surface level transport if that actually fits what you're trying to accomplish. In the previous presentation, we heard something about the eConti project. I'll just say briefly that I'm one of the people at BOKU that is involved in that. So we set up this multi-column chromatography system, and we have a whole strategy for controlling the loading time. And this is just focused basically on the feed zone and how we don't lose product in the flow through. So we have a very simple model to predict how a protein break through. And this looks like this. So this is also essentially based on mechanistics, but it's an analytical solution to the mechanistic equations. So it's very simple and can be computed in Excel. If that's all you need, then why go into more detail? On the other hand, we can also go complex for design. Like here, model every single column, everything outlet, inlet, the protein parameters until you arrive at the simulation of elution of the whole process. So what I want to talk about is in mechanistics, you can, most of the time, it does not work out of the box, and you have to identify the goal or the problem that you're doing for model calibration and changing your mechanistic models around. So big question is, and it's always, is it a transport problem or is it a binding problem? Or in the worst case, it's kind of both. But in this case, I will present two case studies of work that I've done here at BOKU with chromatography modeling. One will be on a transport problem, and one will be on a binding model problem. So in the first case study, I will present a transport phenomenon we observed when we incubated affinity resins with sodium hydroxide, which resulted in a mass transfer change. So if I can show you here that this is a protein A resin, we did it for MAP Select Sure and Prisma. And you can see here that the breakthrough curve gets steeper. So this indicates that the mass transfer gets faster after incubation with sodium hydroxide. The question is now why? We did several experiments, and we saw through confocal laser scanning microscopy that there seems to be some kind of change of the type in diffusion. Now this is where I said, yeah, maybe I can simulate this, and can we verify actually that the diffusion type changes, and can we quantify the change? So for people not familiar with the experimental part, I will just briefly talk about the visualization of single particles with confocal laser scanning microscopes. So you fluorescently label your protein and incubate it with chromatography resin. And you take a sample over time and go down to the particle level and look at the migration of the adsorption front inside the particle. So you can actually see how your protein is moving over time inside a single particle. This looks like this. So in red, there's the protein. In this case, of course, a monoclonal antibody. And you see over time, it migrates towards the center in what we call a shrinking core fashion. And my task now was to recreate the exact conditions in silico of this experiment and adjust mass transfer parameters so we can see exactly what changed in the pre and after the incubation of the resin. I'll show you now an example of a single particle level simulation for the pure diffusion type. So on the right-hand side, you will see solid diffusion. On the left-hand side, you will see pore diffusion. And you should probably look at the solid diffusion first because that is much faster. You can see really the distinction between them. So in solid diffusion, the adsorption front is very diffuse, whereas in the pure pore diffusion, for affinity resins especially, because the binding is very strong, is this shrinking core type. So you can actually visually identify what type of diffusion is prevalent in your system. And the result of this, you can see on the left-hand side here before incubation, is the image I just showed you. And for all of these, we had the actual experimental diffusion coefficients. So in the first step, I just recreated in silico the experiment and just assumed pure pore diffusion. And you can see this matches up quite well to the experimental images. And in the second part of this graph, you can see what happens after the incubation. So you see it's migrates faster for sure, compare this depth to this. And it's also slightly more diffuse, so there must be something different. And we also measured a higher diffusion coefficient. So first of all, this would be what it would look like with just pore diffusion, so we're nowhere close to the experimental images. And then once we increase the contribution of the solid diffusion, we get slowly more diffuse until we arrive at a pretty good match to the experimental conditions. I should mention that these are linked together to form the actual experimental diffusion coefficient. So this is equal to both of these. So in this case, we can actually verify that the diffusional change happened in the type of diffusion and actually quantify it just by modeling it. So this would be then interesting if you have some kind of transport phenomenon. In the second case study, I will be presenting a binding capacity issue where we have a deviation from the expected competitive Langmuir behavior. I'll introduce first our model system. We have a two-component modeling system with adsorption and displacement, so co-adsorption of conalbumin and GFP. And the model proteins, conalbumin or overtransferrin with a PI of 6.8 and GFP with a PI of 5.6. On an anion exchange resin, GFP binds more strongly, and in co-adsorption, it displaces conalbumin. So more GFP will bind the conalbumin, and the conalbumin will have to migrate out of the chromatographic particle. And this we checked out with a system of two separate resins with very distinct structural features. So porous 50HQ is a polystyrene resin with very large pores, up to 1.6 microns. And Q-Sapphirose fast flow is an agarose-based resin with very narrow pores of about 60 nanometer. So our expected behavior of these resins would be they have in the large pores, you have fast diffusion, so including also the displaced conalbumin. So once it's displaced, this has no problem migrating out, and we expect a displacement in column chromatography, like in a breakthrough, a very sharp breakthrough curve, and there's a clear separation between the two proteins. And in Q-Sapphirose fast flow, in the narrower pores, we expect the same behavior that GFP displaces conalbumin, but due to the diffusion limitations, the curves will be much more shallow and the separation will not be as complete. So far for the model system. But for the binding model, we first tried just a standard fully competitive Langmuir model. And we tried the approach that we solely use single component data we obtained by doing isotherm experiments and breakthrough experiments with the proteins separately, and then predict how they would behave in a mixture just with a multi-component Langmuir model. And the idea behind this is that this is a pore. All ligands are available to both during loading, and once it's full, GFP displaces the conalbumin according to the selectivity, and then conalbumin has to diffuse out alongside the concentration gradient. And depending on the pore size, this is either fast or slow. So far for the setup. So we did single component experiments and just try to do predictive modeling with a very simple Langmuir model. So here you can see the experimental results overlaid with essentially the prediction. So here in the solid lines, there's the model, and the dashed lines are experimental. Or in this case, the scatter is of course the experiments. See, the isotherms look quite good. We just, again, used single component data and just plugged it in. This is pretty okay. So this looks good, no problem so far. In the breakthrough curves for Q-Sephiroth fast flow, we can see in the shape is quite correct, but there are some differences in capacity maybe, and maybe also in the equilibrium constants. So the overshoot could be higher, the shape could be a little bit different, but overall, I think can agree that the model is not terribly wrong for this type of system. So with the minor adjustments like capacity, we can actually get to a result. But here, with porous 50 HQ, we have a problem. So again, these are the experiments, and this is the model. So we have a very big problem because GFP capacity is overestimated by about 50%. So we're nowhere close to the capacity obtained. But why does it work in the isotherm data? So we thought, okay, it must be some kind of difference between these two materials. And if you look at porous 50 HQ in a transmission electron microscope, you can see very large pores, but also very narrow and constricting pores. So we thought about it and we said maybe because of this bimodal nature that has large pores and very small pores, maybe not all binding sites are created equal in this resin. So we have a 2D approximation of a macropore and a micropore. So basically to include the structural information of porous compared to Sephiroth into the binding model. So we will have two separate binding sites, one in the macropore and one in the micropore. And the micropore, because of maybe kinetic limitations, it is not as reversible, this the binding, as it is in the macropore. So the idea was we have a reversible competitive binding site in the macropore region, but we have an irreversibly kinetically limited binding site here in the micropore. So whoever comes first stays there, basically. This would also explain the difference between the isotherm and batch uptake kinetics, where both of the proteins have access to both a macro and micropores at the same time, versus in the binary breakthrough experiment, where conalbumin is displaced and the pure conalbumin band is moving downstream in the column, it gets to these small cracks first. And then after when the GFP comes, it cannot enter this because it's kinetically limited, and over the time scale of the whole breakthrough curve experiment, it is not able to displace the conalbumin that has been deposited basically in these micro cracks. So, and with this idea behind, we had these equations, we easily done. Basically, it's just a removal of the ability to desorb from these micropores. And with that, we have a model that matches basically all of our experimental results. You can see here, this matches quite well. For this, I'm actually pretty sure that these 2 points are experimental errors, because especially this one goes beyond the measured single component maximum binding capacity. So I think this is for sure an error here, because in all the other experiments that are easier to perform than a multi-component isotherm, in binary batch uptake kinetics, where I have a lot of protein solution and just put resin in there and just measure, we have a very good agreement over time of the GFP and the conalbumin in solution, or I guess here on the resin. And the binary breakthrough is also solved. So you can see here the capacities are totally in line once we have this 2D approximation of the actual structure of the resin included in our binding model. Sorry. So these were the two case studies where I just want to show you how we use chromatography modeling at BOKU, mostly. I used it for now. And if you are interested how to get started with modeling, we are in cooperation with the people at Jürgen. So I learned Cadet when I was in my master thesis. If you've never heard about it, Cadet is a chromatography simulator environment. So it's a core process that is very modular. You can build complete actor systems in this with CSTRs and all of the things you need. And there's inverse problem fitting in there, so you can have your data, you can have parameter estimation, you have predictive modeling. It's basically a toolbox to get you what you need. And the most important part for university, it's absolutely free. And you can just get started when we close this webinar, essentially. You can just go to the site, download it. The only thing is you have to know Python. There is no graphical user interface. There are different levels to the thing. So it's essentially there's a core that's programmed in C, but you have to access it and build your models. You have to have the syntax of the Python implementation of Cadet, and for data export, you also will need Python. They're working all the time on making it more user-friendly. So there's something called Cadet process, which makes it much easier to create even complex models and connections between 2 columns, 4 columns. The SMB I did, it was basically very easy. And yeah, so far for the Cadet. So I can only recommend it if it is very modular. You can do anything you want. They're even doing compartment models of transport processes in plants if you want to. So it's really a toolbox to do whatever you want with it. And to that end, I was informed that the Cadet workshop is coming up, and you can see all of this. I can talk to you about it later. You can register. There are only 30 participants in the workshop. You get access to the people who develop Cadet, and the tutorial is very good. If you have never done anything with it before, it goes from building a first model, doing parameter estimation, optimization, or if you are already experienced, they also offer problem-oriented solutions just for you. And if you have time and want to register, I can put the link up later in the other session, or you can even try to do it yourself, since the workshop content is also free here on GitHub. As an other option for people in industry or people who don't want to have additional burden of learning Python and just want to get working, there's also GoSilico chromatography modeling software. I've never worked with it, but I've heard it has a very sophisticated graphical user interface. It's more geared toward industry. So you can also have pre-characterized chromatography columns directly from Sativa. They offer tailored solutions and client training, since it's since you have to pay for it for the software. There's discounts for students and teachers, and I believe the focus is really more on usability. So you cannot do compartment models of transports in plants in GoSilico. It's really chromatography focused and application focused, I would say. So with that, I'm at the end of my presentation, and thank you for your attention.
Thank you, Jürgen. Really great talk. Yeah, this brings us to the end of Jürgen's talk. There is one question already in the Q&A. Maybe you want to answer. You see it, Jürgen? I can read.
I can only do it when I stop sharing, I think.
Can you elaborate a bit on how you match the predictions to the experimental results obtained by visual inspections? This is about slide 12.
Slide 12. Okay. Was it the CLSM?
This is the CLSM
right.
of the.
Let me check the slide.
Yeah.
I don't think. Okay, right. I can do the.
Yeah. Maybe there are more questions. Yeah. So yeah. So how did you match the predictions with the experimental observations? Is the question.
Yeah. In essence, it's really the constricting factor that we had was that we had to somehow match the experimental value there's this correlation where you have a combination in parallel diffusion where you have both pore and solid. So I only had a range to work in. Actually, what I did is basically you can get the fluorescence spectrum out of this via, for example, Fiji. You can read it out or ImageJ.
Yeah, and any image processing.
Any image processor, you can get the fluorescence band out. And this was the one that within the constraints actually matched that the best. And then I hope it's on top. Can I get this?
I hope this answers the question.
Yeah,
maybe.
I hope.
So if you, maybe we can have a follow-up question. Are there any more questions to Jürgen? If not, then yeah, I thank you. I thank everyone for joining. I hope today we could show the various different applications for hybrid and mechanistic models for filtration and chromatography for downstream applications. I also want to invite you for the next webinar, which will be on metabolic and cellular modeling. We will have speakers from the Institute of Microbiology and Microbial Biotechnology at BOKU University, and also from the Biochemical Network Analysis Group at the University of Vienna. And if you are interested in talking about modeling and simulation in the framework of this webinar, please write me an email. We still have slots remaining for webinars this year. And if you want to talk to Jürgen, Max, or myself, there is a link to a breakout session in the chat. You can just meet us there. And with that, we can close on time. Yeah, there are no further questions. So thank you, and yeah, I maybe see some of you in the breakout room. Bye.
Bye.
Thank you. Bye.